So in this video, we're going to look at the concept of hemopoiesis, which is basically a fancy way of saying making red blood cells. And to give you an idea, this is a very busy process in that we're not talking hundreds, we're talking billions of cells that have to be made on a regular basis. Now, hemopoiesis is the production so hemopoiesis, there's a term, is the production of blood, especially its formed elements. We're not going to spend too much time on the really detailed embryological development, but basically in the embryo, the yolk sac produces stem cells, which become the first blood cells, and they colonize the fetal bone marrow, the liver, the spleen, the thymus. And many of these sites are involved in hemopoiesis. The liver stops producing red blood cells at time of birth. The spleen will stay involved with lymphocyte productions. Now, the red bone marrow produces all 7 of the formed elements. And the basic pathway is we got something called pluripotent stem cells. And they're the ones that will give rise to the other ones. Then what we have is we have a series of differentiations and different paths that the cells go down to get to the final cell. We have colony-forming units. We have myeloid hemopoiesis. So myeloid, when we think myeloid, really think about the term bone marrow. We have lymphoid hemopoiesis, which is blood formation, lymphatic organs. So we can subdivide them based on those two things. And you already know what the functions of red blood cells are. They're there to carry oxygen, CO2, etc. Also, the blood itself is carrying a number of other things, the nutrients, the water, yada yada, and also proteins and various nutrients. Just to talk a bit about a red blood cell, its design is fabulous. We're looking at here, so there's no sharp edges as long as it's a healthy red blood cell. We're not looking at something like sickle cell anemia. You can also see by having this biconcave shape, it really gives a fairly large surface area for the volume of the cell. And we know from many parts of this course that a large surface area basically allows you to basically do a lot of very rapid exchange. It also allows the red blood cells to be a little bendy McTwisty because they're going to have to go through very small capillary spaces. To give you an idea, we're looking at 7.5 micrometers diameter, about 2 micrometers in thickness. And when mature, they basically lose nearly all their organelles. It's kind of wild that they carry oxygen, yet they lack mitochondria, and they actually rely upon anaerobic fermentation. But that's okay because they have a large surface area and they're bathed in blood, so they can deal with the buildup of lactic acid, and it won't build up. And they also lack nucleus in DNA, which of course indicates that they cannot repair themselves. Now, it's a pretty tough life as a red blood cell, constantly going through blood vessels through the heart, etc. And they really don't have the ability to repair themselves, so they have a limited lifespan. The blood type of a red blood cell is determined by basically markers. There's markers on its surface, and that the shape of the markers basically determines what the blood type will be, and we'll have a look at that. So it really comes down to things like the glycoproteins and the glycolipids. There are also cytoskeleton proteins, which make sense because if you're going to try and maintain the shape, you're going to need a fairly extensive cytoskeleton to help you do it, and allows you to bend and stretch. Now, we've talked about gas transport before. I'm not going to spend much time on this, just to point out that the increased surface area to volume ratio really allows for a lot of exchange. About 1/3 of the cytoplasm is occupied by hemoglobin. And I wouldn't ask you to memorize this number, but give you an idea. But there's about 280 million hemoglobin molecules in one red blood cell. And they're about O2, CO2. And we've already talked about carbonic anhydrase in the lung chapter. Let's have a quick look at hemoglobin. Hemoglobin is actually basically a big conjugate type protein. You can actually see that here's one chain. So I have a chain of proteins there. I have a second one, and you notice these first two gold ones, this one and this one, they're the same type of protein. So there's two copies of it. And then there's another pair of proteins that are similar. So really it comes down to it's four proteins, and two of them are what we call the alpha, and two of them what we call the beta. Now, associated with this, we have a heme group. There's a heme group right there. And in the heme group, so we're going to talk about heme groups right here, it's non-protein that we have in the middle, which is basically ferrous iron. So it's an Fe2 at the center, and we can see it right there. So it's quite a complex structure, and you can have a look at the molecular model down here. And you can see that, if I look at here and expand it, you can see there's the iron sitting right in the middle of that group. Now, we already mentioned the term hematocrit. It's basically the packed cell volume. What are some standard numbers? For men, it's about 42 to 52%. For women, it's about 37 to 48. There's a lot of variability. If a woman, living at high altitude, you'll have higher density of red blood cells. If you're a trained athlete, you'll also have that. I'm not going to spend much time on the numbers for the hemoglobin concentration or the red blood cell count. But if you look overall, the trend is that the values are slightly lower in women on average. Now, of course, there's a lot of exceptions to this rule. A couch potato man versus a very physically fit, aerobic, active woman, right? Makes a big difference. But why are some of these differences? One is that the androgens tend to stimulate red blood cell production a little bit more. There's also the argument that during reproductive years that the loss of blood during periodic menstrual flow you'll lose amount of blood. And also there's sort of this relationship that hematocrit is inversely proportional to percent body fat. Those are some of the explanations given, but I think there's a lot of other things that mitigate that, as I said. Living at sea level versus the altitude, how physically fit you are, etc. And that, could have a large effect. Not going to spend a whole lot of time going through the details of all the steps, but we can quickly see that we start off as a pluripotent stem cell. Then with the right chemical signaling, we become a colony-forming unit. And from there, signaled on to become a erythroblast. And we're not quite at a red blood cell yet, but we're getting there. And then it ends up as, remember, we often go from a blast to a site. And in this case, we go to a reticulocyte. And you can actually see that there's sort of this spotted material in there. And from there, reticulocytes then eventually become the mature erythrocytes, reticulocytes to erythrocytes. To give you an idea, this is a very quick turnover. There's about 2.5 million red blood cells are produced per second. The average life is about 120 days because it's a pretty tough life and you can't repair yourself. Development takes 3 to 5 days. You have to change the cell quite a bit. You got to develop the cytoskeleton. You got to get synthesis of hemoglobin, loss of the nuclei. As we said, the first committed cell is the erythrocyte colony-forming unit. And it's a cell that has receptors for something called EPO, erythropoietin. And erythropoietin is a hormone made from the kidneys that will stimulate red blood cell production. And erythroblasts will multiply and synthesize hemoglobin. And when we get to a reticulocyte, what's happening? The nucleus is gone. Now, how does it get its name, reticulocyte? That tends to refer to the mesh, reticulo. And what we see here, it's a fine network of the endoplasmic reticulum, and that will eventually disappear. Now, if you actually take a blood sample, you'll see that a small percent of the cells in a person's blood are the newly released reticulocytes, and it makes up a small percent, from 0.5% to just over 1%. Just want to talk quickly about iron metabolism because, of course, we know that Fe2 is important. Now, one of the problems is humans are not very good at ingesting iron. And our iron is a mixture in our diet of Fe2 and Fe3. Now, what happens is when that mixture comes into the stomach, one thing that stomach acid can do is convert the Fe3 to an Fe2. Now, iron is so precious, and we're really not that great at absorbing iron, so we really look after it. And what happens is the Fe2 binds onto a transport molecule, gastroferritin. Think of it as like basically an Uber driver. It will pick it up and hold on to it. And that way, when it gets carried, almost chauffeured into the small intestine, in the small intestine, the iron is released and can cross into basically the blood at that point. Now, the carrier molecule, which is the gastroferritin, okay, does not get absorbed. And look at the name, gastro for stomach, ferritin for iron, right? So the name makes sense. Now, there's another Uber type of company that picks up the iron once in the blood, and this is called transferrin. Okay, so trans for transport. That's why I like to think of it. And it'll carry it and distribute it throughout the body. One place it can end up is it can end up in the liver, and it gets dropped off at the liver. And here you see these triangles. Those are ferritin, or apoferritin, I should say. And the apoferritin will bind with the iron, and I end up with ferritin. And that becomes a storage form of the iron. Now, as I said, we're very poor at maintaining our iron levels, is that we have a daily loss in the urine, the feces, and through bleeding. And in men, it's about 0.9 milligrams a day. In women, about 1.7 milligrams a day that we need for just a replacement. But because the absorption rate is so low that we actually have to consume much more than that. So we have to consume about 5 to 20 milligrams a day. So we have to consume this amount just to replace those amounts. And as I said, we've already talked about it. The Fe3, much of it gets converted to Fe2. Gastroferritin binds it, then transferritin will transport it, and apoferritin and ferritin in the liver. Now, there are some nutritional needs for erythropoiesis, and some of the big ones are vitamin B12 and folic acid. These really help with cell division and with DNA synthesis because it's so rapid we need it. Also, vitamin C and copper, they're cofactors for enzymes that help with the synthesis of the hemoglobin. Okay, I'm going to end it there, and we'll pick up the next one.